You can eat clean, take your daily walk, drink your water, and sleep a full 7 hours. And while you're doing all of that correctly, a separate hormonal system buried deep in your abdomen is actively working against you. It has been doing this for years. Nobody told you about it. Not your doctor, not the fitness magazine at your gym, and certainly not the wellness industry, which generates hundreds of billions of dollars every year, selling products to people whose underlying biology was never once explained to them. By the end of this, you're going to understand exactly why belly fat behaves so differently after 55, not as a vague idea, but as a specific nameable mechanism with a specific nameable fix. I'm going to give you the three-part framework I call the V-Reset Protocol. I'll name it now and explain it at the end because the science that leads into it is the reason it works, and if you skip to the end without understanding the biology, you'll follow the protocol for 2 weeks and quit. First, here's what almost nobody tells you. The belly fat you've been trying to lose is not the same substance you were dealing with in your 30s. It looks the same from the outside. It responds to the same bathroom scale. But biologically, after 55, a growing portion of abdominal fat has transformed into something more complicated, something that is no longer just a storage problem, but an active participant in your metabolism. It is producing hormones, it is generating inflammation, and it is, in a very precise biological sense, fighting back against every attempt you make to reduce it. That is the invisible villain in this story. It is called visceral fat, and it is not the fat you think it is. Before we get into the mechanism, let me strip away the most common assumption people carry into this conversation. When most people think about belly fat, they're thinking about the soft, pinchable layer directly under the skin. You can grab it. Doctors call it subcutaneous fat. This kind of fat is largely passive. It stores energy, it insulates, it responds over time to caloric deficits. It is visible and frustrating, but it is not your main biological opponent after 55. It is the decoy. The real problem lives deeper. Behind your abdominal muscles, wrapped around your liver, your pancreas, your intestines, and your kidneys, sits a different kind of fat entirely, visceral fat. What makes it uniquely dangerous, not in the clickbait sense, in the documented clinical sense, is that it behaves like an organ, not a storage tank. An organ, it secretes chemical messengers. It participates in your hormonal system. It generates chronic low-grade inflammation. Researchers at Harvard Medical School have described visceral fat tissue as functionally similar to a gland, one that secretes compounds that circulate in your bloodstream and reach every organ in your body, including your brain. The specific messengers visceral fat releases are called adipokines, molecules produced by fat tissue. Among the most well-studied are inflammatory compounds including interleukin-6 and tumor necrosis factor alpha, both of which are documented drivers of systemic inflammation. This is not the acute inflammation you get from a sprained ankle that you can see and feel. This is a low-grade, constant, silent burn happening inside your body right now if you carry significant visceral fat. Researchers call it metaflammation, metabolic inflammation, and it is linked to insulin resistance, cardiovascular disease, cognitive decline, and a substantially elevated risk of type 2 diabetes. A large meta-analysis published in the European Heart Journal found that high visceral fat area was associated with roughly a twofold increase in cardiovascular risk, independent of overall body weight, meaning you can have a completely normal BMI, the ratio of your weight to your height, and still carry enough visceral fat to place you in a high-risk cardiovascular category. Cardiologists have a phrase for this. They call it TOFI, thin on the outside, fat on the inside. But here is where the story becomes specifically about you, at this age, after 55. The reason visceral fat accumulates so aggressively in this decade, and the reason it resists standard weight loss strategies so stubbornly, is not a single cause. It is four overlapping biological changes, each one feeding into the next like links in a chain. We are going through each one, not to alarm you, because understanding the chain is the only way to break it. The first change is hormonal, and it plays out differently in men and women, though both converge on the same outcome. In women, the dominant shift is the loss of estrogen after menopause. Estrogen is not primarily a reproductive hormone, or rather, it is not only a reproductive hormone. It also plays a central role in fat distribution. When estrogen levels are high, the body preferentially stores fat in the hips and thighs. Researchers call this the gluteofemoral pattern, and it is actually metabolically protective. Hip and thigh fat in premenopausal women consistently shows lower inflammatory activity and lower cardiovascular risk than abdominal fat. When estrogen drops, that preferential routing disappears. The body shifts fat storage to the abdomen, not because you are doing anything differently, but because the hormonal instruction changed. The study of women's health across the nation, known as the SWAN study, tracked thousands of women through the menopausal transition over many years. Researchers found that women in that study gained, on average, roughly 5 to 8 pounds during the menopausal transition, and that the fat gained was disproportionately abdominal, even in women whose total caloric intake and physical activity remained essentially stable. The same body, different hormonal instructions, completely different fat geography. In men, the equivalent is the slow, decades-long decline of testosterone. After 30, testosterone levels drop at approximately 1 to 2% per year. By 55, a typical man may have 30 to 40% less circulating testosterone than he had at 25. Testosterone does several things directly relevant to this conversation. It supports muscle maintenance, it suppresses visceral fat accumulation, and it governs the ratio of lean tissue to fat mass. As it drops, that balance tilts. Muscle decreases, visceral fat increases. These two processes reinforce each other in a loop. Now, here is the mechanism that most doctors never explain clearly, and the reason I consider it the central insight of this entire video. Visceral fat contains an enzyme called aromatase. This enzyme's job is to convert androgens, the hormone family that includes testosterone, into estrogens. So as visceral fat grows, it becomes a factory that actively converts your remaining testosterone into estrogen. In men, this accelerates hormonal decline and further suppresses the signals that would otherwise promote lean tissue. In postmenopausal women, this creates an abnormal, unregulated estrogen signal, not the healthy rhythmic estrogen of the premenopausal years, but a continuous, low-level estrogen produced by fat tissue itself, which the research literature has linked to a higher risk of certain hormone-sensitive conditions. The fat is not sitting there passively waiting to be burned. It is biochemically active. The more of it you accumulate, the more aromatase you produce, the more testosterone you convert, and the more visceral fat you become biologically primed to add. This is a documented hormonal feedback loop, and it is one of the primary reasons that standard dieting fails so consistently and so frustratingly after 55. This is where the mainstream betrayal in this story happens, and I want to be direct about it. The advice you have been given is not wrong for a 25-year-old. It is incomplete, sometimes dangerously so, for a body over 55. We have been told for decades that weight loss is fundamentally a calorie problem. Eat less, create a deficit, lose weight. Physiologist Ansel Keys tested the logic of this to its limit in 1944 and 1945 in what became known as the Minnesota starvation experiment. Volunteers consumed roughly half their normal caloric intake for 6 months. What Keys documented was not a clean, linear fat loss response. The body slowed its metabolic rate, it dismantled muscle tissue for fuel, it dramatically amplified hunger hormones. More than 70 years of subsequent research confirmed every one of these adaptations. The body in caloric deficit is not a passive system being emptied. It is an active system fighting to survive, and after 55, the weapons it uses to fight back are more powerful than at any earlier decade. The NIH-funded Calorie Trial, one of the most rigorous long-term studies of caloric restriction ever conducted in humans, confirmed that significant caloric restriction in middle-aged and older adults resulted in measurable losses of lean mass, including both muscle and bone, alongside fat loss. And the visceral fat specifically was more resistant to loss than subcutaneous fat, meaning the visible fat you can pinch responds more readily to calorie cutting than the invisible organ-wrapping fat that actually drives your cardiovascular and metabolic risk. Here is the exact mechanism. When you restrict calories significantly, more than around 500 per day, cortisol rises as part of the stress response to perceived famine. Cortisol, as we will discuss in detail shortly, specifically signals visceral fat cells to retain triglycerides. Simultaneously, the reduction in muscle-stimulating hormones during a caloric deficit accelerates sarcopenic muscle loss. You lose weight, but the composition of that weight is weighted heavily toward muscle rather than visceral fat. Your resting metabolic rate drops because muscle is metabolically expensive tissue, roughly 6 calories per pound per day at rest versus 2 calories per pound per day for fat tissue. The visceral fat, partially shielded by elevated cortisol, holds on. And when you return to normal eating, which almost everyone does because sustained severe restriction is biologically untenable for most humans, you regain fat faster than you regain muscle, further worsening the body composition that made visceral fat accumulation likely in the first place. The Framingham Heart Study, running since 1948 and one of the longest cardiovascular studies in American history, found that repeated cycles of this kind of weight loss and regain were associated with worse long-term cardiovascular outcomes than stable, higher body weight. Weight cycling compounds the problem it is supposed to solve. I want to be genuinely clear here. I am not anti-medicine. When my husband came close to dying from a cardiac event about 5 years ago, it was the medical system that kept him alive. I respect that deeply. But what he received afterward was a standard protocol, a full stack of medications, and a handout that said, eat less saturated fat and walk more. Which is not wrong. It is just radically insufficient when you understand the six-layer biology driving the problem. Together, under his doctor's supervision, and with careful attention to what the research actually showed about metabolism in a man over 50, we rebuilt his health in ways the standard protocol alone was never designed to achieve. I say that carefully because results vary enormously, and nobody should alter a medical protocol without working with their doctor. But understanding the mechanism, that part is not optional. That is the foundation. Now let's go to the second major player in this story, cortisol. Cortisol is your primary stress hormone. In short bursts, it is essential. It raises blood sugar for fast energy, suppresses non-essential functions during a crisis, and mobilizes stored fat for fuel. It is a survival tool. The problem is that human physiology evolved for brief physical threats, a predator, a flood, a famine, not for the chronic, low-grade, unresolvable psychological stress of modern life. Financial pressure, work demands, poor sleep, relationship friction, processed food. Your adrenal glands, two small glands sitting on top of your kidneys, cannot distinguish between a charging predator and a stack of unread emails. They produce cortisol for both. The connection between cortisol and visceral fat is direct and mechanistically specific. Visceral fat tissue has a significantly higher density of cortisol receptors than subcutaneous fat. This means visceral fat is more biochemically responsive to cortisol than the fat sitting under your skin. When cortisol is chronically elevated, it signals visceral fat cells to absorb and retain more triglycerides from the bloodstream. It also activates an enzyme in visceral fat tissue called lipoprotein lipase, which actively promotes fat storage at that location. Elissa Appel at the University of California, San Francisco published research in psychosomatic medicine demonstrating that women with stronger cortisol responses to psychological stress had significantly higher visceral fat deposits than women with lower cortisol reactivity, even when both groups had similar overall body weight. The stress response is not just shortening your temper, it is geographically directing your fat storage. After 55, the cortisol problem is compounded by a change in the hypothalamic pituitary adrenal axis, the hormonal control system that regulates cortisol output. Research shows that aging blunts the feedback mechanisms that normally suppress cortisol after a stressor resolves. In younger adults, cortisol spikes and then drops relatively quickly. In older adults, cortisol levels following a stressor take measurably longer to return to baseline. The elevated state persists longer. This is not a character flaw or a stress management failure. It is a documented physiological change in the feedback architecture of the stress response system. If you are over 55, managing daily stressors without sufficient recovery, sleeping fewer than 7 hours, or eating a diet high in refined carbohydrates, all of which independently spike cortisol. You are providing visceral fat cells with the exact hormonal signal they are most sensitive to. You are not failing at weight loss. You are running the biological program that grows visceral fat. The distinction matters because the intervention is completely different from eating less. Sleep deserves its own extended moment here because the research on sleep and body composition is some of the most striking in all of metabolic science, and it is almost never discussed specifically in the context of belly fat. Studies conducted at the University of Chicago Sleep Research Laboratory by Corinne Spiegel and Eve Van Cotter demonstrated that restricting sleep to 5 to 6 hours per night caused an 18% reduction in leptin and a 28% increase in ghrelin. Leptin is the hormone that signals your brain that your energy stores are sufficient and that you have eaten enough. Ghrelin is the hormone that drives hunger. Drop leptin and spike ghrelin simultaneously, and you are neurologically programmed to eat more, feel less satisfied when you do, and store more of what you eat. The subjects in those studies did not choose to be hungry. Their brain chemistry changed. Their hunger was not a failure of discipline. It was a predictable, measurable hormonal consequence of insufficient sleep. After 55, sleep architecture changes independently of behavior. Deep sleep, technically called slow-wave sleep, decreases as part of normal biological aging. Growth hormone, released primarily during deep sleep and central to fat metabolism and muscle repair, declines accordingly. Research published in the New England Journal of Medicine in 1990 demonstrated that growth hormone supplementation in men over 60 produced significant increases in lean mass and reductions in fat mass over 6 months. I am not advocating pharmaceutical growth hormone. The point is that the hormone responsible for much of the overnight repair and metabolic recalibration your body performs is directly tied to deep sleep, and deep sleep is decreasing in a way that is invisible to most people. You fall asleep, you feel like you slept, but the quality of that sleep, the depth of it, is diminished, and the hormonal consequences are real. Cortisol and sleep form a bidirectional loop that accelerates visceral fat accumulation in a way that neither factor alone would predict. Poor sleep elevates cortisol the following day. Elevated cortisol degrades sleep quality the following night by shortening slow-wave sleep and fragmenting sleep architecture. The loop tightens over months and years. And because cortisol is also catabolic, meaning it breaks down tissue for energy, chronically elevated cortisol specifically degrades muscle tissue while protecting visceral fat. You lose the tissue you want to keep and keep the tissue you want to lose. Now let's talk about muscle, because this is the structural center of the entire problem. The medical term is sarcopenia, from the Greek for poverty of flesh. It describes the progressive age-related loss of skeletal muscle mass, strength, and function. Muscle mass typically peaks in the late 20s to mid-30s and begins a gradual decline. After 50, research shows an acceleration, approximately 1 to 2% of skeletal muscle mass lost per year in sedentary adults. By 65, many adults have lost 20 to 30% of the muscle they had at 30, and the loss is not evenly distributed across muscle fiber types. Fast-twitch muscle fibers, the powerful, metabolically active fibers used in strength and explosive movements, decline faster than slow-twitch fibers. This is not a minor biological footnote. Fast-twitch fibers are the primary glucose sinks in your body. When you eat carbohydrates and blood sugar rises, skeletal muscle tissue, predominantly those fast-twitch fibers, is where the majority of that glucose gets absorbed and stored as glycogen. As those fibers atrophy, blood glucose has nowhere efficient to go. It lingers in the bloodstream, drives prolonged insulin release, and when storage capacity is genuinely saturated, gets converted by the liver into triglycerides and directed into fat storage, specifically visceral fat. The interaction between sarcopenia and visceral fat accumulation is documented thoroughly enough that researchers coined a specific clinical term, sarcopenic obesity. A person with sarcopenic obesity may not appear classically obese by standard weight measures. Their BMI may sit in the normal or mildly overweight range, but they have lost disproportionate amounts of muscle and replaced it with fat, particularly visceral fat. This population carries substantially higher risk of type 2 diabetes, cardiovascular disease, disability, and mortality than people who are obese without muscle loss. A systematic review published in Obesity Reviews in 2021 analyzed data from over 30 studies and found that sarcopenic obesity was associated with a 2.4-fold increased risk of metabolic syndrome compared to either condition in isolation. This is why the standard advice, cut calories and do more cardio, is structurally incomplete for someone over 55. Cardio burns calories in the moment, but if you are not simultaneously providing a powerful enough stimulus for muscle maintenance, the caloric deficit draws partly from muscle tissue. You lose weight on the scale, but the ratio of fat to muscle worsens. And visceral fat, which responds poorly to cardio as an isolated intervention, remains largely intact. There is a concept here called anabolic resistance that almost never appears in popular health content, and it is one of the most practically important pieces of information in this video. In a young adult, consuming roughly 20 grams of high-quality protein after exercise maximally stimulates muscle protein synthesis, the process by which muscle repairs itself and builds. After 55, this system becomes less sensitive. Research from McMaster University, conducted by exercise physiologist Stuart Phillips, one of the most cited researchers in protein metabolism, has shown that older adults require approximately 35 to 40 grams of high-quality protein per meal to generate the same muscle-building response, not per day spread arbitrarily, per meal. And specifically per meal because muscle protein synthesis is triggered by the amino acid concentration arriving at the muscle after a feeding event, not by a running daily total. The practical implication is immediate. If you are eating a typical Western pattern, minimal protein at breakfast, a moderate amount at lunch, the majority at dinner, you are triggering a maximal muscle protein synthesis response at best once per day, and on many days, not at all. The two smaller meals do not trigger the process in older adults. The muscle loss is silent and invisible on a bathroom scale, but it is happening in your body right now if your per-meal protein intake is insufficient and your resistance training stimulus is absent or inadequate. There is one more layer to this story, and it is the one that receives almost no attention in mainstream conversations about belly fat: the gut microbiome. Your gut contains approximately 38 trillion microbial cells, a number roughly equivalent to the total number of human cells in your entire body. These microbes are not passengers, they are metabolic participants. They break down certain dietary fibers into short-chain fatty acids that feed the cells lining your intestine. They regulate the structural integrity of the intestinal wall. They communicate with your immune system. And they influence insulin sensitivity, appetite regulation, and fat storage in ways that were not understood even 15 years ago. After 55, microbial diversity in the gut tends to decrease measurably. Populations of beneficial bacteria, particularly species like Bifidobacterium and Akkermansia muciniphila, decline with age, while the composition shifts toward species associated with higher systemic inflammation and metabolic dysfunction. A landmark analysis published in the journal Cell in 2015 examined gut microbiome data from thousands of individuals and found that microbiome diversity was strongly correlated with metabolic health markers and that this diversity was declining in parallel with the metabolic deterioration seen in midlife. Akkermansia muciniphila specifically is the subject of a genuinely striking body of emerging research. This bacterial species lives in the mucus layer of the intestinal wall and appears to play a central role in metabolic regulation. A human clinical trial published in Nature Medicine in 2019 found that Akkermansia supplementation in overweight adults improved insulin sensitivity, reduced total cholesterol, and was associated with decreased visceral fat accumulation over 12 weeks. The mechanism is compelling. Akkermansia helps maintain the physical integrity of the intestinal lining. When that lining is compromised, a condition formerly called intestinal hyperpermeability, bacterial fragments called lipopolysaccharides enter the bloodstream. These fragments trigger a significant inflammatory response. That inflammation, as we established earlier with visceral fat's adipokine production, is one of the core drivers of visceral fat growth and resistance to fat loss. The gut lining is not a peripheral issue. It is part of the same inflammatory architecture. The practical implication of the gut microbiome data connects directly to dietary fiber. A study published in Obesity in 2012, conducted at Wake Forest Baptist Medical Center, found that every 10-gram increase in daily soluble fiber intake was associated with a 3.7% decrease in visceral fat accumulation over 5 years, independent of total caloric intake and physical activity. That is not a weight loss effect driven by fiber making you feel full and eat less. That is a specific reduction in visceral fat accumulation driven by the microbial and intestinal mechanisms that dietary fiber supports. The fiber feeds the bacterial populations that reduce intestinal inflammation, which creates a metabolic environment less hospitable to visceral fat growth. One additional layer deserves attention before we connect all of this into the protocol: the mitochondrial dimension. Mitochondria are the energy-producing structures inside your cells. If you learned anything about biology, you probably remember them as the powerhouse of the cell. What that phrase doesn't convey is that their efficiency and their density in muscle tissue are central to how well your body manages glucose and fat. After 55, mitochondrial function declines in skeletal muscle. The mitochondria become less efficient at burning fat as fuel. They produce more inflammatory byproducts called reactive oxygen species, and the density of mitochondria per unit of muscle, the number of energy factories per square inch, so to speak, decreases, particularly in sedentary adults. A key regulatory protein called AMPK, which stands for AMP-activated protein kinase, but what you need to know is that it functions as the body's master metabolic switch, becomes less active with age. AMPK is what tells your cells to burn fat rather than store it. It is activated by energy expenditure, by caloric restriction, by cold exposure, and by specific forms of exercise. When AMPK activity is high, cells are in a fat-burning, glucose-clearing mode. When it is chronically low, as it tends to be in sedentary, older, metabolically compromised tissue, cells default to storage mode. The mitochondrial decline and the reduction in AMPK activity are not separate problems. They feed each other. Dysfunctional mitochondria produce more inflammatory signals that further suppress AMPK, and reduced AMPK activity fails to trigger the mitochondrial repair processes that would otherwise maintain their efficiency. The specific intervention that most powerfully restores AMPK activity in skeletal muscle is not a supplement, not a drug, and not a dietary trick. It is resistance exercise, specifically the type of muscular contraction that depletes glycogen stores rapidly, the kind produced by lifting challenging loads through full range of motion. This is one of the reasons resistance training outperforms cardio as a visceral fat intervention in the research literature. It not only builds and preserves muscle tissue, it restores the intracellular signaling that tells muscle cells to function as metabolic engines rather than passive structures. Now let me connect all of this into a single picture, because seeing the full architecture of the problem is what makes the protocol make sense. After 55, the belly fat problem is not a calorie math problem. Estrogen decline in women and testosterone decline in men redistribute fat to the abdomen and reduce the muscle mass that would otherwise metabolize glucose efficiently. The growing visceral fat mass produces aromatase, which further depletes testosterone and creates an abnormal estrogen signal, perpetuating its own hormonal environment. Chronic cortisol, from inadequate sleep and unresolved daily stress, tells visceral fat cells to grow and resist mobilization. The loss of muscle reduces resting metabolic rate and glucose clearance capacity, increasing the flow of triglycerides into visceral storage. The gut microbiome, degraded by both age and the ultra-processed food environment, allows inflammatory bacterial fragments to amplify the metaflammation that visceral fat is already generating. Mitochondrial decline and reduced AMPK activity convert muscle cells from fat-burning engines into metabolically passive tissue, and conventional caloric restriction, applied without addressing any of these underlying drivers, removes more muscle than visceral fat, worsening the exact composition problem that initiated the cycle. Every standard weight loss product is aimed at one layer of this. The biology is 7 layers deep. I do not find this enraging. I find it genuinely absurd that this information is distributed across 6 academic disciplines and never synthesized for the person sitting in a GP's office being told to watch what they eat. The studies exist, they are published, they just live in journals that nobody has time to translate. Here is the V-Reset protocol. 3 components, each one targeting a different layer of the mechanism we just walked through. The V stands for visceral, because this protocol is not designed to help you lose weight generally. It is designed to shift the specific hormonal and metabolic environment that generates and protects visceral fat. The first component is the resistance anchor, not cardio. Resistance training and not light, high-repetition work that the fitness industry calls toning. The research is unambiguous. The training stimulus required to reverse sarcopenia, restore AMPK activity in muscle tissue, and reduce visceral fat specifically is progressive overload. This means training at approximately 65 to 85% of the maximum load you could lift once on any given movement. Training 2 to 3 days per week, targeting all major muscle groups, and increasing the challenge over time. A study published in Obesity in 2012, conducted by researchers at Duke University, found that resistance training alone produced greater reductions in visceral fat than aerobic training alone over the same time period. Not more total weight lost, but meaningfully more visceral fat reduced. The mechanism is specific. Resistance training increases the glucose uptake capacity of fast-twitch muscle fibers, reduces insulin resistance directly, and produces a post-exercise hormonal environment, a growth hormone and testosterone elevation that directly counters the hormonal drivers of visceral fat accumulation. The most common mistake people make with this component is inconsistency. 3 sessions in the first week, soreness, scale back to once a week, then cessation. Research on resistance training adaptations in older adults shows that consistency over 3 to 6 months is the operative variable. Twice a week, every week, for 6 months produces results that 3 times a week for 3 weeks, followed by 2 months off, does not produce. The second component is the protein anchor. Based on the anabolic resistance research from Phillips and others, the practical target for adults over 55 is 35 to 40 grams of high-quality protein at each main meal, not per day spread arbitrarily, per sitting. High quality in this context means proteins that contain all essential amino acids in sufficient quantity, with specific attention to leucine, the amino acid that acts as the primary trigger for muscle protein synthesis. The minimum leucine threshold to maximally stimulate muscle protein synthesis in older adults appears to be approximately 2 to 3 grams per meal. Leucine-rich sources include eggs, dairy, meat, poultry, fish, and certain plant protein combinations. This level of protein per meal also produces a secondary effect. High-protein meals significantly blunt post-meal blood glucose spikes, which reduces insulin demand, which over months reduces insulin resistance, itself one of the primary downstream consequences of the visceral fat cycle. The common mistake: front-loading protein at dinner while barely eating protein at breakfast, under the assumption that the day's total is what matters. Muscle protein synthesis is triggered meal by meal. A 5-gram protein breakfast followed by a 40-gram protein dinner is not metabolically equivalent to 2 meals of 25 grams each. The morning opportunity is wasted, and muscle protein synthesis goes unstimulated for the entire first half of the day. The third component is the cortisol anchor. This is the one most consistently dismissed as a lifestyle soft skill rather than a metabolic intervention, and that dismissal is a mistake supported by nothing in the literature. You cannot out-train or out-eat a chronically elevated cortisol environment. The 3 specific interventions with the strongest evidence for reducing cortisol and improving deep sleep architecture are the following. First, a hard sleep window of 7 to 9 hours with consistent bedtime and wake time, including weekends. The research on what is called social jet lag, sleeping significantly later on weekends than weekdays, shows it produces cortisol and metabolic disruptions physiologically comparable to shift work, including measurable increases in visceral fat accumulation in long-term observational data. Second, eliminating caffeine after noon. Caffeine has a half-life of approximately 5 to 7 hours in most adults over 50, meaning that a 2 in the afternoon coffee still has roughly half its caffeine active in your bloodstream at 7 in the evening. More importantly, caffeine reduces slow-wave sleep depth even when it does not visibly delay sleep onset, meaning you may fall asleep at the usual time and feel like you slept, while your deep sleep was blunted and your growth hormone release was suppressed. Third, a parasympathetic activation practice in the evening. Parasympathetic means the rest and digest arm of your autonomic nervous system, the physiological opposite of the fight or flight state that cortisol activates. The research supports specific practices: slow diaphragmatic breathing for 5 to 10 minutes, non-competitive low-intensity movement such as a 30-minute evening walk, or progressive muscle relaxation. These are not soft wellness suggestions. They are documented autonomic nervous system interventions. They lower cortisol, improve sleep architecture, and shift the hormonal environment away from visceral fat accumulation and toward recovery. The common mistake is treating sleep and stress recovery as afterthoughts, things to address once diet and exercise are optimized. The data inverts this completely. Cortisol management is not downstream of the other interventions, it enables them. A person performing resistance training and eating adequate protein while sleeping 5 hours a night is still running chronically elevated cortisol, which blunts growth hormone response to exercise, limits muscle protein synthesis during overnight recovery, and keeps visceral fat cells in active storage mode. The three components only work together. The V-reset protocol taken as a whole is not additive, it is multiplicative. Resistance training restores insulin sensitivity and preserves the fast-twitch muscle fibers that absorb glucose. Adequate per-meal protein provides the amino acid substrate that the resistance training stimulus demands for muscle repair, while independently improving glucose metabolism. Cortisol management through sleep and evening recovery unlocks the hormonal environment that allows the first two components to function as intended. Remove any one leg and the other two are measurably less effective. One last thing before the close, because I want you to understand why this is not just a vanity conversation or even just a cardiovascular risk conversation. The inflammatory compounds that visceral fat generates, interleukin 6, tumor necrosis factor alpha, and others, cross the blood-brain barrier. Research from the University of California, Los Angeles, published in Brain Behavior and Immunity, found that elevated visceral fat was associated with measurable reductions in gray matter volume and reduced connectivity in memory-related brain networks, independent of age, total body weight, and cardiovascular risk factors. The belly fat accumulating in your abdomen right now is reaching your brain through your bloodstream. It is generating the same chronic metaflammation in your neural tissue that it generates in your vascular tissue. This is not a future risk, it is a current process, happening silently, without symptoms, until the symptoms are not reversible. That is the mechanism. That is the full picture. And that is why the V-reset protocol is built around biology, not around a number on a scale. If you are going to start the V-reset protocol this week, comment the word reset below so I can see who is actually doing this. And subscribe, because next week I am going into the specific resistance training protocols designed for adults over 55. The actual loads, the actual movements, the frequency, and the three mistakes that turn strength training from a visceral fat reversing intervention into a cortisol spike that makes everything worse.